Students will be able to study about insects fossils and fossils preservation. Students will understand the essential characteristics of freshwater and marine insects fossils. Students will be able to make study on compressions, impression and mineralization. Students will be able to understand evolutionary history in Devonian, Carboniferous, Permian, Triassic, Jurassic, Cretaceous, Paleogene, Neogene in different periods. Students learn about phylogeny, evolutionary relationships, taxonomy, early evidence, origin of insect flight and different theories. 1.2 Introduction. The evolution of insects was based on studies of the following branches of science: molecular biology, insect morphology, paleontology, insect taxonomy, evolution, embryology, bioinformatics and scientific computing. It is a recent understanding. The estimation of the class of insect originated on Earth approximated 480 million years ago, in the Ordovician, in the same time of terrestrial plants appeared. Evidences occurred that the insects may have evolved from a group of crustaceans. The primitive insects were landbound, but before 400 million years ago in the Devonian period one lineage of insects evolved flight. The oldest insect fossil has been proposed to be Rhyniagnatha hursti, estimated to be 400 million years old, but the insect identity of the fossil has been decided. Worldwide climate conditions changed several times during the past of Earth, and along with it the variation of insects. The winged insects pterygotes underwent a major radiation in the Carboniferous, 356 to 299 million years ago, while the insects that go through different life stages with metamorphosis, Endopterygota, underwent another major radiation in the Permian, 299 to 252 million years ago. Survivors of the mass extinction at the Permian-Triassic boundary evolved in the Triassic to what are essentially the modern insect orders that persists to modern times. Insect families of most modern time appeared in the Jurassic, and further diversity probably in genera occurred in the Cretaceous. It is believed that by the Tertiary there existed many of what are still modern genera, hence most insects in amber are indeed member of extant genera. 2. 1.3 Origin of insects. There is a Cambrian explosion before 500 million years ago, a remarkable evolutionary event. During this period the emergence of a plethora of life forms, some gigantic, other small, some that lived on land and other in water. Giant insects, some with wingspans of 2.3 feet, inhabited the Earth made their first appearance before 412 million years ago. Biologists analyze that insect become top predators for nearly 100 million years ago. The most devastating mass extinction period means Permian-Triassic (PT) extinction event that the Earth has ever witnessed. Nearly 90% of all life forms extinct. These extinctions continued for nearly 12 million years, around 251 million years ago, and the insects took a massive beating. 8 orders of insects were completely extinct and 10 insect orders lost a majority of their species. Evolutionists believe that 30% of the insects were lost during this period and some without even leaving a trace of their existence. The surviving orders, however, adapted for surviving and evolved to occupy every possible nook and corner of the planet. Most modern-day insects originated after Permian-Triassic (PT) extinction period. The parallel survival of plant species further assisted their expansion. Together, insects and flowering plants populated the Earth. And now, once again the insects are being challenged by what evolutionists call the 6th extinction event. 1.4 Evolution. Insect evolution is characterized by rapid adaptation with selective pressure exerted by environment, with rapid adaptation being furthered by their high fecundity. It appeared that rapid radiation and the appearance of new species, a process that continues to this day, result in insects filling all available environmental niches. 1.4.1 Evolutionary history. The insect fossil record found back of 400 million years ago to the Lower Devonian. The pterygotes winged insects faced a major radiation in the Carboniferous period. The paragotes insects faced a major radiation and the Endopterygota faced another major radiation in the Permian period. Modern insects order re-originated and survived after Permian and Triassic 3. Extinction. More modern insect families appeared in the Jurassic, and further variation and diversification probably in genera occurred in the Cretaceous. By the Tertiary, there existed many of what are still modern genera, hence, most insects in amber are, indeed, members of extant genera. Into essential modern form most insects diversified in only about 100 million years. Insect evolution occurred due to height fecundity and environmental pressure created. The insect regular filling the all availability in environmental niche and changes accordingly. The insects regularly changes itself according to the changes in flowering plants and other vegetation present around it. It is due to dependent of insects on their environment. The evolution of insects is closely related to the evolution of flowering plants. Insect adaptations include feeding on flowers and related structures, with some 20% of extant insects depending on flowers, nectar or pollen for their food source. This symbiotic relationship is even more paramount in evolution considering that more than 2/3 of flowering plants are insect pollinated. Which insect have to created the diseases on animals, they are responsible for the decimation or extinction of some mammalian species. 1.4.1.1 Devonian. 419 to 359 million years ago was a warm period without any glaciers is known as Devonian period. The first insects probably appeared in the Silurian period, and probably insects got wings in Devonian according to the phylogenetic study. The fossil records are not well known about of this. The fossils that were considered as Devonian insects, such as Rhyniagnatha hursti or Strudiella devonica, are studied that affinities as insects are insufficient. The entomologists found the first complete insects in the late Devonian period, 425 to 385 million years ago. This observation reduces a previous gap of 45 million years in the evolutionary history of insects. In this period the insect's body segments, legs and antennae are visible, however genitalia were not preserved. Both the above-mentioned species represents new species. Insect without wing but it may be a juvenile. 1.4.1.2 Carboniferous. 359 to 299 million years ago is famous period for its wet, warm climates and extensive swamps of mosses, ferns, horsetails, and calamites is known as Carboniferous. Glaciations of the Carboniferous continued into the Permian and because of the lack of clear markers and breaks, the deposits of this glacial period are often referred to as Permo-Carboniferous in age. Due to cooling and drying of the climate, Carboniferous rainforests have collapsed. All remaining of insects are scattered throughout the coal deposits, particularly of wings from cockroaches deposits in particular are in France. The earliest winged insects are from this time period Pterygota, including the aforementioned Latodia, Colonerodia, primitive stem group Ephemeropterans, Orthoptera, Paleodictyopteroidea. In 1940, the largest complete insect wing ever found is a fossil of Meganeuropsis americana, Fig. 01. Juvenile insects are also known from the Carboniferous period. Fig. 01. Meganeuropsis americana, fossil member. Restoration of Mesotheros, a Carboniferous member of the now extinct order Paleodictyoptera. The primitive Blatopterans had a large, disciform pronotum and leathery forewings with a distinct cup vein, a unbranched wing vein, lying near the claval fold and reaching the wing posterior margin. These were false characteristics of cockroaches, as they had an ovipositor, although through the Carboniferous, the ovipositor started to diminish. The previous orders Colonerodia and Myomoptera recently known, with Orthoptera and Latodia to be among the earliest Neoptera which is evoluted from the upper Carboniferous to the Permian. These insects had wings with similar form and structure, small anal lobes. Species of Orthoptera, like grasshoppers and related and adaptive for jumping, hind legs is preserved. 1.4.1.3 Permian. The Permian 290 to 245 million years ago, during which all the Earth's major land masses were collected into a single supercontinent known as Pangaea. At the end of the Permian, the biggest mass extinction in history occurred, collectively called the Permian-Triassic extinction event. 30% of all insect species became extinct. This is one of 3 known mass insect extinctions in Earth's history. 2007 study based on DNA of living beetles and maps of likely beetle evolution 5. Indicated that beetles may have originated during the lower Permian, up to 299 million years ago. During this time, many of the species from the Carboniferous diversified, and many new orders developed, including Proplethroptera, primitive relatives of Plecoptera, Paraplecoptera, Socoptera, Mecoptera, Coleoptera, Rhaphidioptera, and Neuroptera, the last 4 being the first definitive records of the Holometabola. 1.4.1.4 Triassic. A period 252 to 201 million years ago is known as the Triassic. During the Triassic, almost all the Earth's land mass was still concentrated into Pangaea, was when arid and semi-arid savannas developed and when the first mammals, dinosaurs, and pterosaurs appeared. The climate was hot and dry, forming typical red bed sandstones and evaporites during the Triassic. There is no evidence of glaciations at or near either pole. In fact, the polar regions were apparently moist and temperate, a climate suitable for reptile-like creatures. There is only little fossil record of insects including beetles from the lower Triassic. However, there are a few exemptions, like in Eastern Europe entire specimen of the infraorders Archostomata, i.e., Atomocinidae, Schizocolidae, Adephaga, i.e., Trichlidae, Trachypatchidae, and Polyphaga, i.e., Hydrophilidae, Burridae, Elateroidea, and in nearly a perfectly preserved condition. However, species from the families Cupetidae and Schizoporoidae are not present at this site, whereas they dominate at other fossil sites from the lower Triassic. Around this time, during the late Triassic, Mystophagous, or fungus-feeding species of beetle, i.e., Cupetidae, appear in the fossil record. In the stages of the upper Triassic, representatives of the Algophagous, or algae-feeding species, i.e., Trichlinidae and Hydrophilidae, begin to appear, as well as predatory water beetles. The first primitive weevils appear, i.e., Obrienidae, as well as the first representatives of the rove beetles, i.e., Staphylinidae, which show no marked difference in physique compared to recent species. This was also around the first time evidence of diverse freshwater insect fauna appeared. During the Triassic, Hemiptera included the Cercopidae, the Cicadelidae, the Sixidae, and the Membracidae. Coleoptera included the Carabidae, the Staphylinidae, and the Trachypatchidae. Hymenoptera included the Xylidae have appeared. Thysanoptera first appeared as well as Diptera included the Anisopodidae, the Chironomidae, and the Tipulidea are of some of the oldest living families appear. The first true species of Diptera are known from the Middle Triassic and Late Triassic. 6. 1.4.1.5 Jurassic. There is warm and humid climate in Jurassic period. It is during 201 to 145 million years ago. During this period it was important in one of the insect's major predators and the development of birds. Approximate 150 important sites with beetle fossils are in many important sites in Eastern Europe and North Asia. In North America there are only a few sites with fossil records of insects in the Jurassic period. In Asia and Europe numerous deposits of other insects are occur. During the Jurassic there was a dramatic increase in the known diversity of family-level Coleoptera. The development and growth of carnivorous and herbivorous species includes in this. Close to the Upper Jurassic, decreased the copepod portion, however increased the diversity of the early plant-eating, or phytophagous species at the same time. Most of the recent phytophagous species of Coleoptera feed on flowering plants or angiosperms. 1.4.1.6 Cretaceous. Approximately 145 to 66 million years ago the Cretaceous had much of the same insect fauna as the Jurassic not after. 1/3 of Earth's present land area was submerged at the peak of the Cretaceous transgression. It had been seen in the last epoch of the Jurassic in which the Berriasian epoch showed a cooling trend. Snowfalls were common in the higher latitudes and the tropics became wetter than during the Triassic and Jurassic, it is evidence. From the Cretaceous there are a large number of important fossil sites worldwide containing beetles. During the Cretaceous, most of them are located in Europe and Asia and belong to the temperate climate zone. During the Cretaceous, the diversity of Cupetidae and Archostomata decreased considerably. There is distribution of different pattern of predatory ground beetles, Carabidae, and rogue beetles, Staphylinidae, whereas in the warm region the Carabidae predominantly occurred. Preference of many areas with temperate climate, the Staphylinidae and click beetles, Elateridae, are found. Likewise, there is hunted their prey under the bark of trees together with the jewel beetles, Buprestid, by predatory species of Claroidea and Cucujoidea. The primary consumers of wood, the jewel beetles' diversity increased rapidly during the Cretaceous, while longhorn beetles, Cerambicidae, were rather rare and their diversity increased only towards the end of the Upper Cretaceous. 7. 1.4.1.7 Paleogene. From this era there are many fossils of beetles known, though the beetle fauna of the Paleocene, 65 to 56 million years ago, is comparatively poorly investigated. Beetle fauna is very good in contrast. The knowledge on the Eocene, while there are many fossils of beetles known from this era, though the beetle fauna of the Paleocene is comparatively poorly investigated. The reason is the occurrence of fossil insects in amber and clay slate sediments. Amber is fossilized tree resin. That means it is not consists of minerals, it is consists of fossilized organic compounds. Location, age and species of the resin-producing plant, distinguish the different type of amber. Amber is most important for the research on the Oligocene beetle fauna, Baltic and Dominican. The first butterflies are from the Upper Paleogene, while most, like beetles, already had recent genera and species already existed during the Miocene. However, their distribution differed considerably from today's. 1.4.1.8 Neogene. The Neogene, 23 to 0 million years ago, is most important sites for beetle fossils are situated in the warm temperate and to subtropical zones. During the Miocene many recent genera and species already existed, however, the distribution differed considerably from today's. The most important fossil sites for insects of the Pliocene is Willershausen near Göttingen, Germany, with most preserved beetle fossils of various families, longhorn beetles, weevils, ladybugs and others, as well as representatives of other orders of class insect. Well-known fauna of beetle is in the Pleistocene, since the composition of the beetle fauna has been used to reconstruct climate conditions. 1.5 Phylogeny. We believe is the world's oldest known full-body impression of a primitive flying insect, a 300 million-year-old specimen from the Carboniferous period. Millipedes and centipedes are closely related to crustaceans. Millipede, centipedes, scorpion and spider are sometimes confused that they are very similar to insects with their body plans. From the 396 million-year-old Rhynie-chert fossils is the oldest definitive insect fossil is the Devonian Rhyniegnatha hursti. It is superficially resembled a modern-day silverfish, order Thysanura, insect. This species already possessed dicondylic mandibles, 2 articulations in the mandible. It is a wingless insect but have a 8 character associated with winged insects. It is evidence that wings may already have evolved at this time. In the Silurian period, it seemed that the first insects probably appeared earlier. There have been 4 super radiations of insects. Beetles evolved around 300 million years ago, flies evolved around 250 million years ago, moths and wasps evolved around 150 million years ago. These 4 groups account for the majority of described species. Mecopteri evolved the flies and moths along with the fleas. The origins of insect flight remain not well known, since the earliest winged insects currently known appear to have been capable flyers. Some extinct insects had an additional pair of winglets attaching to the first segment of the thorax, for a total of 3 pairs. There is no evidence that suggests that the insects were a particularly successful group of animals before they evolved to have wings. The record of insect fossil extends back some 400 million years to the Lower Devonian, while the pterygotes, winged insects, undergo a major change in the Carboniferous. The Endopterygota undergo another major change in the Permian. In the Jurassic period most modern insect families appeared, and further modification probably in genera occurred in the Cretaceous. In essential modern form the insects have diversified in only 100 million years. The insect have evolutes due to rapid adaptation by selective pressure of climatic conditions and their high fecundity. Regular adaptations result was new species formation. This process was continued till now, its result that the insects filling all available environmental niches. Approximately 20% of extant insects were depending on flowers, nectar or pollen for their food source, and the symbiotic relationship is even more paramount in evolution considering that more than 2/3 of flowering plants are pollinated by insect. The extinction of some mammalian species was done by some particularly flies and mosquitoes which was the vectors of some pathogens. 9. Fig. 02 left traditional phylogeny of insects reflecting the uncertain placement of key groups with aquatic larvae blue, an inferred ancestral state in the common ancestor of Pterygota, the winged insects. Tree topology integrates over a number of historical phylogenies, based on morphological data and/or Sanger sequenced loci. Right phylogenomic tree of a resolved Polyneoptera, red, from the Supermatrix analyses of Wipfler et al. 2019 reflecting the ancestral state reconstruction of an aquatic larval stage. The insect's phylogeny is the sequential study that underlies various macroevolutionary phenomena, such as the winged flight evolution, Fig. 02 parental care, interaction study with plants, and morphological specializations for diverse modes of feeding, specialization of mouth 10 parts. However, a stable insect phylogeny has historically remained, but now it is basis on molecular systematic nearly 2 decades ago. 1.6 Summary. The animal groups and the evolutionary relationships of insects are not clear, although there have been many number of opinions expressed. Few evolutionists explain that the arthropods are a phylogenetic group and that of many similarity has been found as a result of convergent evolution, however few have explained that it is monophyletic, having evolved from a common ancestor. There is 5 major theories of the ancestry of insects exist, with one or more leading scientists explain for each. They include the speculation that the ancestor common to all insects was an annelid or annelid-like form that looked something like a modern earthworm. Class Insecta is also explained by many scientists to have evolved from a myriapod millipede, or some type of proto-myriapod animal during the Devonian period, but even this conclusion about insect origin is controversial. The segment worms apparently evolved from the ancient protozoans concluded by Meglich and other, which also gave rise to the mollusk and were the ancestors of insects. Labandera explain on the basis of bimolecular studies that insects evolved from an unspecified lineage of crustaceans. They admit that insect origins remains unsettled and that morphological evaluations still favor a non-crustacean ancestor. The most common theory is that insects evolved from some type of myriapod. Myriapods and insects both evolved from some unknown common ancestor, it is concluded by Romazer and Stoffolanon. Some evolutionists explain that it is near to suppose that the earliest insects were similar to silverfish. Apites in the fossils record an estimated 350 million years ago. The rarity of transitional forms is the major reason why phylogenetic trees vary so drastically. The dominant view of insect evolution, from myriapods, is not supported by RNA analysis. The limbs of crustaceans are branched by ramus, it is one major difference, whereas those of insects and myriapods are always unbranched, even in their embryonic stages, thus their new name Uniramia. The mouthparts of Chelicerate also have unbranched set, in particular a pair of pincer-like structure called chelicerae. Finally, there are fundamental difference between the group in the 11 way the legs and jaws move, which suggests that each developed limbs independently. Of the ancestors themselves, little is known, and only the worm-like onychophorans provide any real evidence. The construction of either body wall and excretory system is distinctly annelid-like, but they have appendages also that could have been the forerunners of the insect's segmented limbs, as well as insect-like antennae, and trachea for breathing. All this suggests that the Uniramia evolved from annelid-like, but the ancestors from the other groups, the crustaceans and chelicerates, remain a complete mystery. The great advantage of using insect fossils to test evolution is that most insects have exoskeletons, and consequently their external morphology is usually well preserved. 2.1 Objectives. To study insect classification up to families. To study phylogeny of Arthropoda and Insecta. To study the Apterygota, primitive insects. To construct the dichotomous key for insect identification, to study how to identify the insects with the help of dichotomous key. 2.2 Introduction. Insecta is the largest class of phylum Arthropoda, it including nearly millions known species, about 3/4 of all animals, in which an estimated 5 to 10 million undescribed species. Insect body divided into 3 parts: head, thorax which bears 3 pairs of legs and usually 2 pairs of wings, and many segmented abdomen. Many species undergo incomplete and complete metamorphosis or metamorphosis absent. Insecta are divided into 2 subclasses: Apterygota primitive, wingless forms, including silverfish and bristletails, and Pterygota, more advanced winged or secondarily wingless forms. The approximately 29 orders of Pterygota are generally classified by wing form, e.g., Coleoptera beetles, Diptera, Dipterans, Heteroptera, bugs, Lepidoptera, Hemiptera, etc. Insects are found in almost all terrestrial and freshwater and some marine habitats. On the basis of wings present or absent, insect divided into 2 subclass, Apterygota and Pterygota. Apterygota subclass further divided into 4 order on the basis of tail structure, and Pterygota subclass divided into 2 division. Division 1 is Exopterygota and division 2 Endopterygota on the basis of wing develop externally and internally respectively. Exopterygota division further divided into 16 order and division 2 Endopterygota divided into 10 orders. Total orders are 30 according IMS classification. 15. 16. Subclass Apterygota, Ametabola, wingless subclass Pterygota, Metabola. Winged insects. Division: Exopterygota, Hemimetabole oa. Wings develop externally. 1. Protura telsontails. 2. Dipleura diplurans. 3. Collembola springtails, Snowfleece C4. Thysanura bristletails, silverfish at C5. Ephemeroptera mayflies 21. Neuroptera antlion sandlace wings 6. Odonata dragonflies sandamselflies 22. Macoptera scorpionflies. 7. Plecoptera stoneflies 23. Trichoptera caddisflies. 8. Dictyoptera cockroaches sandmantids 24. Lepidoptera mocksandbuttflies. 9. Gryloblatodea gryloblattids 25. Diptera flies, mosquitoes 10. Orthoptera grasshoppers, locusts 26. Siphonoptera fleas 11. Crickets, mole crickets at sea 27. Hymenoptera ants, bees, wasps 12. Phasmida stick insects 28. Coleoptera beetles, weevils 13. Dermaptera earwigs 29. Strepsiptera stylopids 14. Embioptera web spinners. 15. Isoptera termites. 16. Zoraptera zorapterans. 17. Socoptera booklice. 18. Melophaga bird lice. 19. Siphunculata anaplura sucking lice. Class Insecta. IMS classification. G. RAGHAVAI. AH. Closing parenthesis. 17. 20. Hemiptera plant bugs 21. Thyssanoptera thrips Pteractosoft subclasses. Apterygota, Ametabola, Pterygota, Metabola. Primitive insect S and small insect S advance and most developed insect S. Most liwingless wingless mouth parts are encloses or hidden. Int he head. Mouth H parts exposed or externally visible. Mandibles monocondylicichh articulate with head at a single point. Mandibles dicondylICWHICH articulate with head at 2 points. Excretory organs is malfigant oobles WHICHARE Arab center rudiment ARY. Era present. Only in adult. (2.4) Phylogeny of Arthropoda and Insecta. Phylogeny of arthropods. The monophyletic theory is proposed by Snodgrass, 1938, Sheriff, 1966, and they suggest that all arthropods have evolved from a single annelid-like ancestral stock whose limbs are lobopod-like. This lobopod annelid gave rise to Tetracephalosomida and the most primitive arthropods. Trilobitomorpha evolved from the Tetracephalosomida, and Chelicerata and Mandibulata originated from Tetracephalosomida. Onychophora evolved from lobopod annelid-like ancestor and represent an early lateral branch of the evolutionary line. The monophyletic theory is based on the segmentation between the annelids and arthropods, and the prostomium and pygidium of the annelids correspond to the acron and telson of arthropods. A number of anatomical features, such as chitinous cuticle, hemocoel, dorsal blood vessel, segmental jointed appendages and centrolecithal eggs constitute the basis for the view that the arthropods are monophyletic. Anderson, 1998. The polyphyletic theory is proposed by Teagues and Manton, 1958. They suggested that there were two ancestors of arthropods. The lobopod annelid is considered to be the ancestor of Onychophora, which was originated in the Precambrian period about 525 million years ago. The group Uniramia, which includes chilopods, diplopods and insects, is thought that they have evolved from the base of onychophores. The proto-annelid ancestor is considered as the primitive annelids from which the most primitive extinct arthropod group Trilobitomorpha arose, from which two extant groups, Crustacea and Chelicerata, evolved by two separate evolutionary lines. Manton, 1973, Ruppert and Barnes, 1994, support the polyphyletic theory. They suggest that three different living groups, Chelicerata, e.g., horseshoe crabs, spiders, scorpions, etc., Crustacea, e.g., copepods, barnacles, prawns, crabs, etc., and Uniramia have evolved separately, possibly from the annelid ancestors. This view is based on the locomotory structures and embryonic development of the different major groups. 25. Insect affinities have long been controversial. There is two putative sister groups being Myriapoda and Crustacea, but the recent morphological and molecular advances support a Crustacea clade. The monophyletic theory of the Insecta is also well supported. It is a very ancient group, as the oldest records are from the early Devonian. Among animals, arthropods have been considered to be earliest colonizers of land based on fossil evidence. However, it is possible that other animal taxa, e.g., nematodes, tardigrades, and annelids grouping land even earlier, but these groups have relatively poor fossil records. Nevertheless, they are far from being the oldest terrestrial animals, as the first traces of a possible terrestrial arthropod are from the Cambrian or Ordovician of Australia. Arachnids and myriapods are represented to Ordovician and Silurian terrestrial arthropods. This hypothesis is not supported by the fossil record of the early based on taxa described from very few Devonian outcrops. These Devonian Apteris s belong to two main lineages: the Entignatha colembula, Protura, dipleura, with mouth parts hidden in the cephalic capsule, and the Insecta, a clade that comprises the two Apteris orders Archaeognatha and Zygentoma, plus the winged forms are Pterygota. Zygentoma and Pterygota are characterized by dicondylic mandibles. All these clades are known in the Devonian or the Carboniferous and are still living, except perhaps the controversial order Monora that comprises the sole extinct family Dasyleptidae, recorded from the late Carboniferous to the Triassic. This situation is extraordinary compared to the fact that many of the early Paleozoic major vertebrate clades are now extinct: Thelodonti, Osteostraci, Placodermi, Embolomery, etc. Even the oldest Collembola reniella precursor has a very modern habitus similar to those of the modern springtails. The situation completely changed after the early Carboniferous with an explosion of diversity that affected the winged insects, but not the Apteris lineages. The oldest known outcrops with pterygoda are dated from the Namurian of Germany, Czech Republic, and China. These localities provided very rich and diverse entomopaunas, with species belonging to extant taxa cockroaches, Orthoptera or grasshoppers, Odonatoptera or dragonflies, etc., but also other that correspond to extinct, strictly Paleozoic groups as Paulida and Neopteran group dominating in the earliest late Carboniferous deposits, and the most famous one being the Paleodictyoptera. Our knowledge on these earliest winged insects has greatly increased during these last years. 2.5 Introduction to primitive insects. 26. 2.6 Construction of dichotomous key for identification. A dichotomous key is a technique of identification whereby groups of insects are divided into two categories repeatedly. With each continuous sequential division, more information is revealed about the specific features of a particular insect. When the insect no longer shares its totality of selected characteristics with any insects, it has been identified. When using a dichotomous key to identify insect specimens, it is preferable to use immutable features, i.e. features that do not change. Size, shape, coloration, color pattern, habit, habitat and behavioral patterns may all vary amongst individuals and across lifetimes. Physical structures, e.g. number of legs, prolegs, number of wings, antennae shape and size, and biological processes, e.g. reproduction methods, type of genitalia, type of respiration, structure of spiracle, make for better characteristics. Dichotomous keys are usually represented in one of two ways. As a branching flowchart, diagrammatic representation. As a series of paired statements laid out in a numbered sequence, descriptive representation. 2.7 Summary. It is unclear the relationships of insects to other animal group. There is the more traditionally grouped with millipedes and centipedes. The closer evolutionary evidence ties with crustaceans has emerged in favor. The insects together with Remipedia and Mollucostraca according pan-crustacean theory make up a natural clade. Such as centipede, millipede, scorpions and spiders. Some terrestrial arthropods body similar to some insects are confused. The first insect was origin earlier in the Silurian period. It have been 4 super radiations of insects: beetle 250 million year ago, flies 500 million year ago, moths and wasps 150 million year ago, 27. These groups for the majority of insect species. Flies, moth and fleas evolved from the Mecoptera. Large number of successful insect groups has evolved in cooperation with flowering plants, a powerful combination of co-evolution. Some modern insect genera develop during the Cenozoic era. 3.2 Introduction. Insect collecting refers to the collection of insects. Most insects are small and these cannot be identified without the minute morphological characters, so entomologists often make and maintain insect collections. The collections are conserved in research institutions or universities or colleges where they are maintained and studied by specialists. Many college courses curriculum require students to form small collections. Historically, insect collecting has been widespread and was a very popular educational hobby. Slow-active insects are caught using funnels, pitfall traps, bottle traps, malaise traps, flight interception traps and other slow-active of insect traps, some of which are baited with small bits of sweet foods, such as honey. Different designs of ultraviolet light traps such as the Robinson trap are also used by entomologists for collecting nocturnal, night-active insects, especially moths, during faunal survey studies. Pooters or aspirators suck up insects too small or delicate to handle with fingers. Different types of nets are commonly used actively to collect insects. Flying insects are used to collected by aerial insect nets. The bag of a butterfly net is generally constructed from a lightweight mesh due to minimize damage to delicate butterfly wings. A sweep net is used to collect insects from grass and brush. Sweep net is similar to butterfly net, except that the bag is generally constructed from more hard material. The sweep net is swept back and forth through vegetation, quickly turning the opening from side to side and following a shallow figure 8 pattern. During sweeping, the collector walks forward while, and the net is moved through plants and grasses with force. This requires a heavy net fabric such as sailcloth to prevent tearing, although light nets can be used if swept less vigorously. Continuous sweeping for some distance 31. And then the net is flipped over, with the bag hanging over the rim, caught the insects until they can be released by a pooter. Many types of nets used for collecting insects include beating nets and aquatic nets. 2 leaf litter sieves are used by coleopterists and to collect larvae. Once collected, a killing jar is used to kill required insects before they damage themselves trying to escape. However, killing jars are generally only used on hard-bodied insects. Soft-bodied insects, such as those in the larval stage, are generally fixed in a vial containing an ethanol and water solution. 3. Another, now mostly historical, approach is caterpillar inflation, where the innards were removed and the skin dried. Insects and mites of all kinds may be killed and preserved in liquid agents or a dry gaseous agent. Some kinds of insects are best kept dry. Ethanol, grain or ethyl alcohol, mixed with water, 70% to 80% alcohol, is usually the best general killing and preserving agent. For some kinds of insects and mites, other preservatives or higher or lower concentrations of alcohol may be better. Because pure ethanol is often difficult to obtain, some collectors use isopropanol, or isopropyl alcohol. Isopropanol does not seem to harden specimens as much as ethanol. The specific concentration to use depends on the kind of insect or mite to be preserved. 3.3 Collection of insects. Mass collection devices. Light trapping. Mass collection devices light trapping. 32. Fig. 01. Light trap. We take a bucket and make 4 holes at the top rim and make 1 larger hole at the bottom rim. Adjust small size light shade on the bucket in inverse direction. Fix 4 rods with screws, nuts with the bucket for holding up the large size light shade. Fix the light source as shown in the figure. Hang the light trap, Fig. 01, on the large size light shade. Drainage hole on the bucket should be kept closed by the rubber plug, while at the bottom of bucket we take swab with chloroform. Light attracted insects come toward light and capture in bucket bottom and killed by chloroform. Collect, drain out trapped insects periodically. Pinned, spreading, drying process takes place for further study. The light trap installing near or within the field where the maximum diversity occur. Secure the poles firmly on the ground. Approximately the electric bulb on the frame just above 5 meters from the ground. It assure that the electric bulb and wiring are not in contact with water to avoid electrocution. Put the light trap from early evening 6 p.m. to 10 p.m. Collect the trapped insects daily and separate the useful insects for study and useless insects, dispose them properly. Flight intercept traps. Flight intercept traps, FITs, also known as barrier traps, are passive traps that are used to collect insects flying through an environment. Many insects, when encountering a barrier while flying, will either fly up to go over the barrier, or drop down to the ground. This behavior can be exploited to collect insects. Ground level FITs consist of a vertical barrier, collection containers, and a horizontal rainfly, flight intercept trap, Fig. 02. The vertical barrier is typically made of fine mesh, such as mosquito netting, although it could be made of clear plastic or glass. The rainfly is typically plastic. It obviously functions to keep rain from flooding the collection containers, but also functions as a top barrier to reduce escapes, thus enhancing trap success. 33. Fig. 02 Flight intercept trap. Opening parenthesis. App. JPEG collection containers are filled with a killing agent and or preservative agent. Water, with soap to act as a surfactant, may be used as a killing agent, but the traps must be serviced and specimens removed daily, otherwise specimens will begin to rot. Propylene glycol, 6-based antifreeze, 7, or pure propylene glycol, can be used as a killing and preserving agent. Do not use ethylene glycol antifreeze, 8, because of its toxicity to mammals. The sweet taste also increases the likelihood of mammals disturbing the collection containers. If propylene glycol is used, traps can be left for long periods without servicing. Ground level FITs collect an enormous number of specimens over time, 100 plus per day, and will collect numerous non-flying ground-dwelling insects and arthropods as well. Sifting and Burlese funnels. Sifting and Burlese funnels, Fig. 03 and 04, are used to extract small insects and arthropods from leaf litter and other debris, dung, mushrooms, flood debris, etc. A sifter is a device used to concentrate individuals by removing large pieces of substrate. This is done using a container with a wire mesh bottom. The debris is placed in the container, agitated, and the smaller particles and individuals fall through. See sifting, right. If you sift over a white pan, see below, or white sheet, you can immediately begin collecting specimens with an aspirator or brush. 34. Fig. 03 Sifting for insects, Fig. 04 Burlese funnels, separator. Fig. 03 Sifting for insects, Fig. 04 Burlese funnels, separator. Bait traps. Bait traps, FG05, using fruit, dung, carrion, pheromones, heat, color, etc. as attractants, can be general or targeted collection devices. Many bait traps are homemade, while others are available commercially. The general design of a bait trap includes 2 elements. A place to hold the bait. Attractants such as pheromones or colors can be incorporated in the structure of the trap, while fruit, dung, carrion, etc. typically need a cup or compartment in which to be placed. 35. Fig. 05 Bait trap. In southeastern Brazil, adapted from _Fig 2 _236,091,501. In southeastern Brazil, adapted from _Fig 2 _236 mil. Typically this is separate from the compartment used to hold the bait. Another common killing agent, especially for traps that use color or pheromones, is the use of a sticky substance, such as tanglefoot. This is not a preservative, and sticky traps should be checked often if quality, or even identifiable, specimens are desired, and special solvents must be employed to remove the sticky material from the specimens. Pitfall traps. 36. Fig. 06 Pitfall trap. Pitfall trap. A pitfall, Fig. 06 trap, is a simple device used to catch small insects that spend most of their time on the ground. In its most basic form, it consists of a container buried so that its top is level with the surface of the ground. Pitfall traps come in a variety of sizes and designs. They come in 2 main forms: dry and wet pitfall traps. Dry pitfall traps consist of a container, tin, jar or drum buried in the ground with its rim at surface level, used to trap mobile insects that fall into it. Wet pitfall traps are basically the same, but contain a solution designed to kill and preserve the trapped insects. The fluids that can be used in these traps include formalin, 10% formaldehyde, methylated spirits, alcohol, ethylene glycol, trisodium phosphate, picric acid, or even, with daily checked traps, plain water. A little detergent is usually added to break the surface tension of the liquid to promote quick drowning. The opening is usually covered by a sloped stone or lid or some other object. This is done to reduce the amount of rain and debris entering the trap, and to prevent animals in dry traps from drowning when it rains or overheating during the day, as well as to keep out predators. Collection equipment. Nets. Nets used to collect insects and arthropods fall into three general categories: aerial nets, sweep nets, and aquatic nets. Each is specifically designed to interface with the environment in which the sampling will take place. Nets consist of three elements: the handle, the hoop, and the bag. Fig. 07, 08. Fig. 07 Insect net. Opening parenthesis, closing parenthesis. 37. Note on the use of nets: many insects are vigilant. If you intend to sample with a net, do so with competence. Do not blunder around, cast a shadow willy-nilly, shake branches and vegetation, or stomp around in the water or on the bank before collecting. Otherwise your intended specimens will fly, swim away, drop to the ground, or otherwise disappear long before you take a single swipe. Aquatic nets are used to sample aquatic environments ranging from lakes, ponds, rivers, streams, abandoned tires, puddles, tree holes, etc. Aquatic nets range from a standard size, with approximately 1.5 meters, 5 feet long handle, to diminutive sizes, such as aquarium nets with 5 centimeters, 2-inch wide hoops. Standard-sized aquatic nets are robust and can be used somewhat forcefully, but be careful not to rip the bag on hidden underwater debris. Generally, hoops of aquatic nets are flat on the distal, away from the handle edge to facilitate collection against the benthos bottom of the water column. Do not use an aquatic net like a shovel. Scooping too much muddy substrate, gravel, or aquatic vegetation will make separation of specimens from the sample difficult or impossible. After you have collected a sample, it can be washed by bobbing the sample in the water, making sure that the lip of the net doesn't go below the water and allow specimens to escape. Samples placed in a watertight container and kept cool will retain live specimens for hours. Otherwise, samples should be preserved in 80% ethyl alcohol, with replacement 24 hours later if the sample was particularly watery, or can be sorted immediately. Sorting an aquatic sample in the field is best done using a white pan, but care should be taken that small or cryptic organisms are not overlooked. Fig. 08 Aquatic insect net. Opening parenthesis, closing parenthesis. 38. Beat sheets. Beat sheets. Beat sheets are an important way to survey specific plants for insects. Beat sheet. Fig. 09. Foliage should be struck soundly with a stick so insects are jarred loose and fall onto the sheet. Rather than holding the beat sheet horizontal, tip the far corner up. Insects will walk upslope and are less likely to immediately fly away. Specimens are easily collected with an aspirator. Fig. 09 Beat sheets. Opening parenthesis, closing parenthesis. Aspirator. Aspirator Fig. 14, also known as a pooter in entomology, is a device used in the collection of small insects, usually for scientific purposes, for field and lab work. One of the most common designs consists of a small resealable jar or vial, the lid or stopper of which is penetrated by two tubes. On the inner end of one tube, fine mesh or another type of filter is attached, and this tube leads to the user's mouth, usually connected by a long, flexible piece of tubing. The end of the second tube projects into the collecting chamber, and its far end can then be placed over an insect or other small organism. The user sucks on the first tube, and the insect is drawn into the collecting chamber through the other. 3.3.1 Killing jars. Insect kill jars Fig. 10 are an essential piece of equipment for anyone making an insect collection and allow the collector to quickly and easily dispatch the insects they find in the field. While it is possible to make a very simple kill jar that contains only a paper towel soaked in a killing agent, it is nice to have a more permanent jar so you're not getting the fluids all over your bugs. 39. Fig. 10 Killing jar. 3.3.2 Relaxing jars. To make a relaxing chamber Fig. 11, place 1 to 2 inches of clean sand or sawdust in the bottom of a large, airtight jar. The jar must be large enough to allow small dishes to be placed inside, and must have a screw type or other lid to create airtight conditions inside the bottle. Fig. 11 Relaxing jar. 3.3.3 Aerial nets. Aerial nets Fig. 12 are used to collect flying insects such as mayflies, dragonflies, butterflies, moths, true flies, bees, and wasps. Aerial nets are deliberately built light, with long thin handles, sometimes flexible hoops, and a soft bag with a large mesh size. These are ideal for collecting delicate specimens, such as mayflies or butterflies. An aerial net should never contact any solid things, but only scoop the insect from the air. The bag is long so it may be flipped over the 40. Hoop trapping the specimen after a catch. Generally, specimens collected in aerial nets are removed from the net with the hands or transferred from the net to a killing jar Fig. 10. Aerial nets should never be used for beating vegetation or in the water unless you absolutely have to. The only thing worse than ruining a fine net is not collecting a rare specimen. Fig. 12 Extents DP B07JZ5LL7K. 3.3.4 Sweeping or beating nets. Sweep nets FIG13 or beat nets are used for sweeping vegetation or beating dead limbs, etc. They are very robust, with short thick handles, heavy inflexible hoops, and have hardy bags of very small mesh or light canvas cloth. The durability of the net allows the user to assault the substrate with such force that insects are jarred loose and collected in the net. Sweep net samples can be quantified if the area swept per sweep and number of sweeps per sample are held constant. Generally, specimens are collected from a sweep net using an aspirator. If small flies, wasps, or other fast-flying organisms are desired, the entire sample can be dumped into a plastic bag or other container. The specimens can be killed, cooled, frozen, etc. and dealt with later. Sweep nets can be used as aquatic nets in a pinch, although sweeping with a wet bag can be difficult. 41. Fig 13. 3.3.5 The aspirator. Aspirator. An aspirator, also known as a pooter, and more colloquially as a sucky-uppy thing, is a small device used to collect specimens FIG14. A collecting vial is sealed by a stopper that has two tubes. The fixed metal tube is the collection tube and the flexible yellow tube is the suction tube. Notice in the figure's insert that the opening to the suction tube is covered with very fine mesh. This is important. Small insects are sucked through the collection tube and trapped in the collection vial when suction is applied to the suction tube. The fine mesh over the suction tube keeps the specimens from being eaten by the collector. If you use a collection vial with a lid, then multiple collections can be kept separate by capping one vial and getting another. Fig 14 aspirator. Opening parenthesis, closing parenthesis. The collector does not need to fully inhale in order to create proper suction to capture a specimen. In fact, inhaling through the device is not recommended. A kissing technique may be used where air is taken only into the mouth and not into the lungs. Use caution aspirating near fungal spores, dust, or dead animals. Some people attach a new car gasoline inline filter to the end of their suction tube to keep from inhaling dirt and debris. Aspirators can be used for any and all types of collection and should be available whenever one is in the field. 3.3.6 Light traps. Light trapping, ultraviolet lights, black lights, mercury vapor, and various others, is a general term describing the use of lights to collect insects. It exploits some insects' behavior of traveling to a light at night. This is a well-known phenomenon, although definitive explanations of this behavior are lacking. Insects tend to be best attracted to the shorter wavelengths given off by ultraviolet and mercury vapor lights. Trap is a light sheet. This particular example is homemade but contains the basic design elements. The white sheet reflects light and acts as a structure for insects to land on. The sheet can be hung on a tight rope if trees, structures are available. Keep the hanging sheet tight, unlike what is illustrated in the figure, and do not bump it. A sheet on the ground is important as many insects will accumulate there. Mercury vapor top and ultraviolet hanging lights can be used together or in concert. Insects at light sheets are easily collected with aspirators. Different groups of insects tend to come to light traps at different times of the night. For example, bark beetles, Scelidonae, tend to arrive in the early evening, while Saturniid moths, Saturniidae, may not arrive until 1 or 2 a.m. The best strategy is to turn the light on at dusk and keep it on until at least midnight. 3.3.7 Pitfall traps. Pitfall traps, Fig 06, are used to collect ground-dwelling insects and arthropods. The concept is simple. A container with preservative is placed in a hole in the ground and organisms fall in. The best design is a double cup design. After the hole is dug, 2 cups, typically robust disposable plastic drink cups, the expensive brands, are placed in the hole and soil packed around it to the level of the lip of the inner cup. The inner cup can now be removed and emptied while the outer 43 cup keeps the hole from backfilling with soil. It is important that the lip of the inner cup is level with the surrounding soil, otherwise small organisms may see it as a barrier and go around the cup or burrow beneath it. 3.3.8 Pan traps. Pan traps, FIG15, are primarily used to capture Microhymenoptera, but also trap many other insects. All you need for a pan trap is a small colored pan filled with soapy water. In fact, plastic bowls or plates using for picnicking are ideal. Use different colored bowls to catch different insects. Yellow, blue, white, and red are common colors used. To use the trap, just place a bowl on the ground and partially fill with water and a drop of dish liquid or other soap. The dish liquid is used to break the surface tension of the water, so the insects will fall through. Pan traps need to be checked at least daily. To collect the arthropods captured, the water is poured through a fine aquarium mesh net. It is then rinsed with water into a jar of 70% ethanol. Fig 15. 3.4 Preservation. Insect specimens, if properly preserved and cared for, can last hundreds of years. Any collected specimen carries an enormous potential to inform about itself and the time and place of collection. Maintaining any specimen for many years carries a cost. Proper preservation ensures a high-quality specimen, which increases the quality of information the specimen contains, and increases the value of the maintenance of the specimen. Generally, two types of insects preserved on the basis of softness and hardness of the body. The soft-bodied insects preserve in alcohol or 44. Other liquid preservative, while the hard-bodied insect preserve in dry method. Both preservation are as follows. 3.4.1 Soft-bodied insects. Soft-bodied insects should be preserved in plastic or glass vials with alcohol, 70%-85% isopropyl or ethanol alcohol is ideal. These insects include larvae, scale insects, termites, aphids, thrips, silverfish, mayflies and earwigs. Other insects can be preserved in alcohol as well, with the exception of butterflies, moths, and bee flies bombylids because the alcohol will damage their wings. Preserve larval stages of insects and other soft-bodied specimens immediately by dropping them directly into a 70-90% ethyl or isopropyl alcohol solution. When collecting in the field, carry a few small vials of preservative fluid to store specimens. An even better method of preserving caterpillars, grubs, and maggots is to carry them home alive in separate plastic or glass containers, then submerge them for 1 to 2 minutes in boiling water. After this, they can be immediately placed in the vials of alcohol. This process kills bacteria in the digestive tract and prevents discoloration. After a day or two, the liquid may become discolored. When this happens, transfer the specimens permanently to fresh alcohol solution. Specimens in vials must be labeled exactly as described for pinhead specimens with 2 exceptions. Labels must be written with a soft lead pencil or with India ink so that the writing does not dissolve or run. Only one label should be used, thus append any information such as host and identification to the bottom of the label. Cut labels longer to allow for this. Labels must be included with the specimen inside the vial. Do not attach them to the outside as this has proven to be an unreliable method of labeling. 3.4.2 Hard-bodied insects. The hard-bodied insects preserved by dry preservative method. The steps have included in this method is collecting, killing, pinning, spreading, drying and preservation the specimens. The details of this method is as follows. Collection. The insects have been collected by the different method and procedure which explain in above text. 45. Fig 16 Insect spreading board. The procedure is as follows. Pinning insects. Insect pins of various sizes are available from entomological suppliers. It recommend getting size as they're good general purpose sizes for most insects. Use only pins designed for pinning insects as other household pins may rust and damage the specimens. Any insect large enough to be pinned without the risk of breaking or distorting it is pinned from the top, dorsal side, straight down through its body. The correct point of insertion depends on the type of insect or pinning. The insect will be most pliable and easy to pin and preserve right after it dies. Try to pin the insect as soon as possible after it dies for the best results generally. The area is just slightly right of the midline of the insect's thorax. To pin the insect in the recommended location which reduce the risk of damaging important identifying characteristics of the insects. Insert an insect pin through the middle of the thorax, the middle section between the head and abdomen, slightly off center to the right, Fig 17. Push the pin all the way through the insect's body, then about 1/2, into the spreading board or another piece of styrofoam board. The insect should typically be about halfway up the pin or far enough above the spreading board so that you can freely position the legs. After a butterfly or moth is relaxed, carefully hold the specimen under its thorax between your thumb and forefinger. Gently squeeze the thorax, the wings should separate slightly. From the top, insert a pin through the center of the thorax. 46. Figure 17 Pinning method. A fly, B butterfly, C beetle, D grasshopper, E bug, F wasp. Different insects will be pinned. Figure 17 Pinning method. A fly, B butterfly, C beetle, D grasshopper, E bug, F wasp. Different insects will be pinned at different part without damaging its identifying characteristics. 1. Bees, wasps, flies, etc. Pin through the thorax between bases of forewings and slightly to right of middle line. 2. True bugs. Pin through the scutellum, which is the triangular area between the bases of the wings. 3. Grasshoppers, crickets, etc. Pin through the prothorax or saddle, slightly to the right of the center line. 4. Beetles. Pin through the forepart of the right wing cover near the center line. 5. Butterflies, moths, dragonflies, etc. Pin through center of thorax between the bases of forewings. 3.5 Pinning, mounting and displaying of insects. The preferred way to display and study adult insects is to pin them into display boxes using special insect pins. The pins are pushed through the thorax of the insects while they are still fresh and the legs and antennae are placed in a natural position. This allows the specimens to be handled and studied without damaging them. In pinning insects, always use special insect mounting pins, entomological pin, that are specially treated to resist rusting. Insect pins vary in size from 000 to number 8. The latter is the largest in diameter. Numbers 2 and 3 usually work the best for the amateur collector. A good rule is to pin the insect slightly to the right side of the middle and down through the area where the middle pair of legs is attached. Styrofoam is N. 47. Excellent base for positioning the legs so they will dry in the correct position. There is a slight variation in the pinning of the different kinds of insects. Fig 18 Pinning and labeling of insects. Correct pinning. Importante. Do a nice job of pinning insects will require some practice. All insects must be pinned so that they are straight on the pins. Do not tilt the specimens on the pin. The height of the specimen on the pin will depend somewhat on the size of the insect. However, always keep the distances from the top of the insect's body to the head of the pin the same. Usually 1/3 of the pin should be above the upper surface of the insect. This will allow enough room to permit handling specimens without actually touching them. Specimens should be handled carefully to keep all legs and antennae. Fig 18. (48) Fig 19 Different insects pinning method. A small amount of time spent positioning the legs and antennae will dress up the collection. Insect specimens can be kept for long periods of time. Many of the specimens in university collections are nearly 100 years old. Labeling by using pinning block. It can also properly space the labels that go on the pin beneath the insect specimen. The label nearest the insect gives the common name. The second label gives the scientific name. All labels should be of uniform size, white in color, and the information typewritten or printed neatly. The appearance of a collection of insects is important if all the specimens and labels are placed at a uniform height on the pins. This is easily done by using a wooden pinning block. Fig 20. Pinning small insects. Many of the smaller insects cannot be pinned directly through the body with regular insect pins. These should be mounted on card points. Card points are slender triangles of paper. These triangles are pinned through the broad end with a regular insect pin and the insect is glued to the narrow point. Fig 19 and 20. Fig 20 Labeling of insects. 49. 3.6 Summary. Fig 19 Different insects pinning method. A small amount of time spent positioning the legs and antennae will dress up. 3.6 Summary. The collection of insects is an important practices in which the insects identified easily. It is used for study the insects in relation to their beneficial and harmfulness. Insects are collected using funnels, pitfall traps, bottle traps, malaise traps, flight interception traps and other slow active of insect traps, some of which are baited with small bits of sweet foods, such as honey. Different designs of ultraviolet light traps such as the Robinson trap are also used by entomologists for collecting nocturnal, night-active insects, especially moths, during faunal survey studies. Pooters or aspirators suck up insects too small or delicate to handle with fingers. Different types of insects collecting nets are used for this practices. Aerial insects are used to collected by aerial insect nets. Grass and brush insects are collected with the help of sweep net. Sweep net is generally constructed from more hard material. During sweeping, the collector walks forward while, and the net is moved through plants and grasses with force. Continuous sweeping for some distance and then the net is flipped over, with the bag hanging over the rim, caught the insects until they can be released by a pooter. Once collected, a killing jar is used to kill required insects before they damage themselves trying to escape. However, killing jars are generally only used on hard-bodied insects. Soft-bodied insects, such as those in the larval stage, are generally fixed in a vial containing an ethanol and water solution. 70% alcohol is usually the best general killing and preserve agent. Some collectors use isopropanol, or isopropyl alcohol because isopropanol does not seem to harden specimens as much as ethanol. Generally two types of insects preserved on the basis of softness and hardness of the body. The soft-bodied insects preserve in alcohol or other liquid preservative while the hard-bodied insect preserve in dry method. Soft-bodied insects include larvae, scale insects, termites, aphids, thrips, silverfish, mayflies and earwigs. Specimens will be mounted only when they are fully relaxed, i.e. when their legs and wings are freely movable, rather than stiff or dry and brittle. All remounting methods use entomological macropins. These are stainless steel pins. Carding is not recommended for adult insects because structures on the underside are obscured by being glued to the card. However, carding may be suitable for mounting exuvi, pupal cases, puparia, or scale covers. Spreading involves holding the appendages away from the body while the specimens are drying. Legs and antennae can be held in semi-natural positions with pins and the wings can be opened and held out horizontally on a setting board using pieces of tracing paper. 50. (3.6 Summary. The collection of insects is an important practices in which the insects identified easily. It is used for study the insects in relation to their beneficial and harmfulness. Insects are collected using funnels. Different designs of ultraviolet light cellophane, greaseproof paper, etc. Setting boards can be constructed from pieces of polyethylene foam. Drying process completed after 24 hours from spreading. Dry specimens transfer in preserving naphthalene balls fumigated insects box. 3.7 Parental care may be defined as any investment made or any effort taken by parents to take care of their young ones in order to increase the chances of their survival and hence increase their reproductive capabilities. The parents provide food to the young ones, protect them from predators and help them to be successful in life. Parental care is most highly evolved in mammals and bird, but examples of parental care are also found in other animals like fishes, amphibian, insects and other invertebrates. Since insects are a highly diverse group, parental behavior has developed many times independently in response to environmental factors. Insects show the highest diversity with more than 1 million species in the world, but only 1% species have developed parental behavior. Parents of most insects provide almost no care for their offsprings beyond supplying the eggs with a little amount of yolk and placing the eggs in a location safe from environmental hazards. The rare occurrence of post-oviposition parental care in some insects is thought to reflect the early evolution of a complex of traits that effectively protected the eggs and larvae. 53. Stages from predators, parasites and other environmental hazards. So insects provide post-ovipositional parental care that enhances the survival and overall development of the offsprings, often a substantial cost to the parent's future reproductive potential. Most parents provide simple form of care whereby the female attends the eggs until hatching. However, a few insects exhibit elaborate forms of parental care similar to those of birds and mammals. Selection pressure favors lots of offsprings and parental care is limited to species which produce fewer young ones. Parental care can mediate transition from solitary to group of family living both for an individual and on an evolutionary scale. It is likely to have developed as an altruistic trait to enable the parents to better pass on their genes by improving the survival of their offsprings even at the cost of their own energy, food or even future reproductive opportunities. Parental behavior in insects ranges from covering of eggs with protective coating, finding a good habitat for egg laying, forming eusocial societies with alloparental care, feeding and protecting the young ones. Lifelong association of parents and offspring's parental behavior can be observed at the beginning of the egg stage and can last up to the larval stage. Some species, however, put more effort on parental care after the egg hatching stage as larvae are more vulnerable and disposed to predators. Generally, parental care is provided by only females. Maternal care, however, uniparental male care, paternal care, is quite rare. In some cases, biparental care has been seen, i.e., male and female form long association to rear young ones to adulthood. Parental behavior is most developed in the following insects orders: Hemiptera, true bugs; Thysanoptera, thrips; Embioptera, web spinners; Coleoptera, beetles; Hymenoptera, ants, wasps, and bees; Isoptera, termites. 54. 4.2.2 Brood parasitism. Parental care have evolved in some insects from time to time. Brood parasitism is also seen in some insects. It is a reproductive strategy whereby the offsprings of one species are reared in the nest of the same species, interspecific brood parasitism, or different species, intraspecific brood parasitism. Brood parasite manipulates the behavior of the host so that the host takes care of the young ones of the parasites instead of its own young ones. 2. The host is cheated. The cuckoo bees, for example, lay her eggs in the brood chambers of other bees. The young ones of cuckoo bees eat the pollen grains stored in the hive and often eat the eggs of the host species. 4.2.3 Forms of parental care. Parental care is a significant behavioral adaptation in the life history of a number of animals including insects. The forms of parental care are quite diverse, ranging from uniparental care, generally female care, to elaborate and long-term biparental care. Parental care is an ancient behavioral strategy that has occurred independently many times in different insect orders and several other arthropods. It enhances the fitness and survival of offsprings by providing protection from predators, parasites and various environmental hazards as well providing food to the young ones. Maternal care. Maternal care is the most common type of parental care in insects. The female takes care of the eggs and the young one. The simple forms of maternal care provided by the mothers to their eggs is to incorporate some defensive chemical into their eggs, laying eggs in a protected place or covering the eggs with a shell or some waxy substance before